Hello there, I'm Johannes and this is a presentation for STBC3022 Genomics. For introduction, we know that the biofilm formation is vital in bacterial survival strategy, and the focus of the research, which is Burkholderia pseudomallei, is known to produce biofilm. The disease that it causes mitosis is not very difficult to eradicate and its persistence, and thus there is many researchers that is conducted for this. So why do we conduct research in the first place? It's because there's a general consensus after multiple studies involving individual mutants showing that the inactivation of single genes does not completely attenuate biofilm formation. This implies that there is a more global regulation of multiple genes and pathways in biofilm formation in Burkholderia pseudomallei. To validate for that, a comprehensive transcriptional analysis involving both high and low clinical Burkholderia pseudomallei biofilm producers will be conducted. And to do that, we need to first grow them to a mid-log phase, in which we will isolate the total RNA, remove ribosomal RNA, and prepare them to create a cDNA library using NGS. From there, we will do quality preprocessing of the sequence reads, in which we will remove any unwanted sequences and map our required sequence to a reference genome, which is K96243 genome sequence. From there, we will produce a total transcriptome profile, in which we can determine the differential expression of non-transcripts. For downstream analysis, we will conduct hierarchical clustering, p-sort and gene ontology, in which we can conduct them based on Pearson correlation metric and average linkage clustering. Then we will also predict their localization incellularly and also classify them functionally. Because this is transcriptomics, that is why we are dealing with mRNA. These are the slides, please read them. And for results, the research discovered that there are 563 differentially expressed genes that encode for core functions, and these genes have high percentage to be predicted to encode protein that will localize extracellularly. So, what are the genes that have been upregulated? Genes that are upregulated are such as genes that are related to film and pilus were upregulated. Genes that encode for polysaccharides and outer membrane cell surface encoded proteins were also upregulated because they also produce EPS. Genes that are involved in surface composition of the cell is also being altered in high biofilm producers. Other than that, a large R-like domain is also being discovered in Burkholderia pseudomallei biofilm formation, in which large R-like domain is important in quorum sensing and is known to play a major role in the control of bacterial biofilm formation in many known pathogens as well. Other than that, there is also an upregulation of two common signal transcript system and stress proteins, as well as the modulation of the identification enzyme pathway in high biofilm producers of high biofilm Burkholderia pseudomallei producers. From there, we can see that there is a potential correlation between formation of biofilm in Burkholderia pseudomallei and its virulence in nematode and mice models, in which we can see that UM6, which is a high biofilm producer, causes the lesser mean time to death to both C. elegans and bob C mice model. And this trend is not only limited to UM6 or UM1, but also evident in UM5 and UM2, which are high biofilm producers and low biofilm producers themselves. So for discussion, we need to understand how can all these upregulated and downregulated genes actually contribute to biofilm formation, number one, and number two, to virulence. And as we can see in this picture, it shows that in response to very environmental signals such as the presence of nitrate and nitrite, attachment of cellular stress or osmosis shock, Burkholderia pseudomallei will regulate the two common signal transduction systems to adapt to these changes, and to adapt to that, they will transcribe genes that encode for proteins involved in the alteration of surface components and most crucial to initiate attachment of the planktonic bacterial cells, which is Burkholderia pseudomallei, to solid surfaces. So the shift between planktonic form to attachment to solid surfaces requires genes in the first place, and as the biofilm grows, this will induce an oxygen-limited environment in the biofilm itself. So for high biofilm producing Burkholderia pseudomallei strain, they will utilize nitrate for anaerobic growth through the nitrification enzyme pathway. This led to virulence, as the bacteria can effectively live inside the host. So for conclusion, this is the first report of complete transcriptome profile of the Burkholderia pseudomallei biofilm producers, and there's postulation that the ability to sense the anaerobic cues and adapt to them through the nitrification enzyme pathway is pivotal in formation of Burkholderia pseudomallei biofilm in infected hosts that leads to persistent infection. Thus, to solve this problem, no strategy designed to thwart the information is required. Thank you.